Everything so far has been searching, finding molecules like ones we already know. Now we make the jump to prediction. We'll train a model that learns the link between a molecule's structure and its potency, so it can score a molecule nobody has ever measured. It's called QSAR, Quantitative Structure-Activity Relationship, and it's the gateway to machine learning in drug discovery. The idea is simple. Take the fingerprint we've used all series, that's our input, X, 2048 bits per molecule. Take the pick 50 we pulled from ChEMBL, that's our target, Y, show a model enough pairs of the two, and it learns the mapping from structure to potency. Then, for any new molecule, fingerprint it and the model predicts a number. Fingerprint in, potency out. First, features. A model can't read a molecule, but it can read a vector of numbers. So we convert each fingerprint into a number py array and stack them into a matrix, 1065 molecules, 2048 columns each. That matrix X, paired with the pick 50 vector Y, is the entire training set. Now the single most important habit in machine learning, hold out a test set. We set aside 20% of the molecules that the model will never see during training, 852 to learn from, 213 to be judged on. Then we fit a random forest, 300 decision trees, a strong no-fuss baseline for fingerprint data. A couple of seconds, and it's trained. The moment of truth, we predict on those held-out molecules and score ourselves. Two numbers. R², the fraction of the variation we explain, 0.70. And RMSE, the typical error, 0.83 pick 50 units. On the plot, each point is one test molecule, predicted against its real value. The tighter they hug the dashed line, the better. And ours hug it reasonably well. Let's be honest about what this means. An R² of 0.7 and an error near 1 pick 50 unit is a realistic result for a plain fingerprint model, good enough to rank and triage molecules, not good enough to trust to 2 decimal places. That's the true state of QSAR, a fast filter, not an oracle. But a fast filter that scores molecules for pennies is exactly what you want before spending real money in a lab. So we trained a model that turns a fingerprint into a predicted potency, and we measured exactly how much to trust it. This same model will do real work in episode 8, scoring molecules we've never made. But next episode, we leave 2D behind entirely, we load the EGFR protein itself and dock a molecule into its binding pocket in 3 dimensions. See you there.